Welcome, everybody. I see more people are joining in, but again, we are very excited to start the new series of seminars for the Xylella files. This is being organized by Blanca Landa from Spain and Giuseppe Stancanelli from EFSA from Italy, and I'm helping a little bit them. Blanca today is traveling, so she couldn't be here, so she asked me to introduce the speaker today, and I'm very glad to do so. So today we're going to listen from Lena Pesenti. So Lena is in the University of Louvain, Catholic of Louvain in Belgium, when she's finishing her PhD. Hopefully she was telling me soon in January. And under supervision of Claude Bragard. She's been working in both in Xylella for her master's that she's going to tell us a little bit about, and also in her PhD, focusing on the identification of potential hosts in Northern Europe among Salicaceae species. Sorry if I say it wrong. You will correct me later. So these trees act as efficient hosts and possible stepping stones and reservoirs for propagation of the disease in northern regions. So she's been studying how xylem-inhabiting bacterial endophytes influence the outcome of Xylella infections. So her title is How Endophytes Interact with Xylella fastidiosa and Brennerian salicis. So thank you, Lena, for giving this talk, and go ahead.
Okay, thank you for the introduction and the opportunity to speak at this seminar series. So as said, this presentation will develop research done at UCLouvain and will focus on exploring the xylem sap to unravel interaction between bacterial xylem endophyte and two phytopathogenic bacteria, which are Xylella fastidiosa, that I guess you all know, and Brenneria salicis, that maybe some of you are not so familiar with. And we are going to study that in the Salicaceae through metagenomics and in vitro studies. Also, just a note before starting my presentation, you will see during the presentation on the slide different several QR codes that you might want to scan if you want more information about the results that are on the slide, because they are gonna link you to the publication that are linked to the result on the slide. So feel free to scan them if you want more information. So let's begin. The risk posed by xylem-associated pathogen in Northern Europe should not be underestimated. So we are going to take the case, as I said before, of Xylella fastidiosa and Brenneria salicis. If it is well known that Xylella fastidiosa causes major issues in temperate region on plants such as citrus or grapevine, or more recently on olive trees, question remains for Northern European region, which traditionally have cooler climates. However, those regions may become more conducive for the bacterium survival and spread, as we have seen on cooler climates, such as in Canada or Northern USA. So several studies have also shown that the bacterium was able to grow even at lower temperature, and that the climate in northern region could become suitable for its establishment and spread. Xylella fastidiosa is exclusively transmitted by xylem sap feeding insects, and once it's a part of the xylem vessel, Xylella can either degrade the plant wall to move to the neighboring vessels. It can also move up the stream through a process called twitching motility, or when the infectious state is more advanced, it can also form biofilm. Now I'm going to maybe brief you a bit about Brenneria salicis that you might be less familiar with. So Brenneria salicis is a bacterial pathogen that is responsible for the watermark disease in willow trees in region with mild climate, but it's causing significant damage on salix in Europe and other regions. However, the presence of Brenneria salicis is not always linked with disease development, meaning that the bacterium can be described as dormant. The bacteria are thought to enter the tree through wounds, and often it can move in the xylem vessels also. And so the disease is primarily transmitted via contaminated pruning tools or insect vectors, or by the movement or replication of infected plant material. Okay, so with this knowledge gathered on some of the well-known pathosystem, Xylella fastidiosa and Brenneria salicis disease can be studied on Salicaceae, who could play a role of reservoir and stepping stone in northern region of Europe, But what makes Salicaceae efficient host? Well, first of all, they are common in the riparian tree layer, which have already been established as important areas for the development of both disease. They form a close match network in Northern Europe. Those plants can either be symptomatic or stay asymptomatic when they are infected by either bacteria. They also are the host of some Xylella fastidiosa potential insect vectors. This has been determined through a mark release recapture experiment that I will detail later. And both bacterium are able to multiply and disperse in the xylem vessels of the plant. And this has been determined through an inoculation assay that I will also detail later. So we wanted to study the risk in the field. So to do that, we set up a sentinel plantation in the Balearic Island with several tree species, including Willow. So this allowed us to monitor host pathogen interaction in their under natural condition, and to monitor the circulation of the bacterium in the plot and around it. An area was demarcated, and in this area, we did a floristic inventory. We also sampled some insect vectors, and we planted some spy plant. During the 4 years of sampling and monitoring, Xylella fastidiosa was never detected in our sentinel plants or in the collected insect vectors. While it's rather a positive effect not to have any infection of this quarantine pathogen, the duration of the plantation did not allow us to answer the question about the potential host range of Xylella fastidiosa. However, we could still see some symptoms, but they might be due to other factors such as environmental factors, and the lack of result might be due to low vector population. However, this experiment allowed us to have useful information on how to set up sentinel plantation and encourage us to add sentinel plantation in surveillance routine. We would also like to thank the UIB team for helping us in this setup. Then speaking of vectors, local data were lacking to model the potential spread of Xylella fastidiosa in Northern Europe. So the dispersion capacity of some potential insect vector was studied with a mark release recapture experiment that was performed in 2 different sites, one rural site and one urban site, where collected insects were marked with fluorescent powder and then released and recaptured every day for a week and then 1 month later. The other experiment was a flight meal experiment that allowed us to test 2 potential vectors that were Philinus spumarius and Afrophora salicina. And the conclusion to both experiments was that the specialist Afrophora salicina, driven by the distribution of Salicaceae, flies farther than the generalist Philinus spumarius. So now we know more about the dispersion capacity of insect vectors, but what about the transmission capacity? So we also did a transmission test that was conducted on Salicaceae, so on Salix alba and on Populus tremula, where 2 branches per tree were incubated with 5 to 10 insects collected on infected part of the island. And then after the incubation period, we tested the insect and plant sample but by qPCR or by MLST, and several showed acquisition of the bacterium. But we wanted to know more about the propagation of Xylella fastidiosa inside Salicaceae, so we also performed...
Sorry, Lena.
Yeah.
You are not sharing the screen. At the beginning, I was thinking that you were making an introduction, but we are
okay.
seeing your face.
It says that I'm sharing the screen.
Yeah. The screen, I can see the screen. I think.
Okay.
Yeah, I can see the. Yes,
Okay.
I can see the screen also.
Okay, so I changed something. I'm so sorry.
Okay, no worries. Thank you. Yeah, so we wanted to know more about the propagation of Xylella fastidiosa in Salicaceae. And so we did a pinprick inoculation again on some different species of Salicaceae, where 2 branches per tree were inoculated, and then the plant were kept either at 22 or 28 degrees. And we then monitored the propagation of the GFP-marked Xylella fastidiosa with different methods, including confocal microscopy, isolation, PCR on leaves 1, 2, 3, and 6 months after the inoculation, and qPCR on stems 6 months after the inoculation. Symptoms were also monitored every 2 weeks. And if, I don't know if you can see the pointer, but I guess you can. So here you can see the Salix alba and here the Populus canescens, and they are more complex to diagnose visually because we have difficulties to identify clear differences between the control group and the group that are inoculated with Xylella fastidiosa. But if we look at Populus tremula, we can see that there are some clear difference between the group that are controlled, either if they are showing some symptoms because we are in a greenhouses, there are 3, they are in potted in little pots. So yeah, we have still some symptoms on the control group, but they are different than the one on the Xylella inoculated group. And then we also checked the spread of the bacterium with PCR and confocal microscopy. And so in this table, you can see a summary of the detection across the 3 species. So starting with Populus tremula, here you can see that the detection at the inoculation point was always successful. And in most plants, we could also detect the bacteria at the apex and in the roots. And then with PCR on the leaves, we were able to see that the maximum spread from the inoculation point was 65 centimeters above the inoculation point and 70 below. Then for Salix alba, we had an even stronger colonization, and we detected the bacteria at the inoculation point, and then the PCR confirmed a maximum spread of 93 centimeters above the inoculation point. However, by contrast, in Populus canescens, we did not observe significant spread under either temperature, which suggests a much lower susceptibility. And then on those confocal microscopy picture, you can see longitudinal and a transverse section of Populus tremula stems, where you can clearly see fluorescently marked bacteria along the xylem vessels, which are clogging the vessels. And then here for Salix alba, you can see that we still find some bacteria, but they are not clogging the vessels as much. We wanted also to investigate the bacterial loads. This load is expressed in colony-forming units per gram of plant tissue under both temperate greenhouse and tropical greenhouse. And we can see that in Populus tremula, so here and in red on the graph, we consistently recovered very high bacterial population. So this shows that Populus tremula is a very good host. By contrast, on Populus canescens, so here and in yellow on the graph, you can see that we have a much lower bacterial population, indicating only limited colonization. And then Salix alba here and on orange in the graph, it fell in between, so we have consistently colonized, but at a lower level than Populus tremula. We also try to re-isolate Xylella fastidiosa from the infected plant tissue, and you can see how often we were able to re-isolate the inoculating strain from plant tissue. So for Populus tremula, we were able to re-isolate the strain in some cases, both at the inoculation point and at the apex. But in contrast, for Salix alba and Populus canescens, we did not manage to re-isolate the strains under the same condition. So this finding have 2 major implications. The first one is that they confirm that Salicaceae can act as reservoir for xylem-limited pathogen in Northern Europe. And the second one is that it shows that it's really crucial to study not only the pathogen, but also the plant-associated microbiome, because if willow and poplar are permissive hosts, then the bacterial endophytes that naturally inhibit their xylem might play a role in shaping whether the infections can progress or are suppressed. So this is the transition point that motivated us to investigate the xylem microbiome of Salicaceae, and that's what I'm going to describe in the next section. So given those characteristics, it is believed that the xylem endophyte might play a role in mitigating symptoms development in host plants infected with either Xylella or Brenneria. In fact, the microbiome associated to Xylella fastidiosa in some of the host plants is already well studied. Endophyte can suppress pathogen growth through direct microbial interaction or by priming the host plant's immune response. And the idea would be to apply this kind of knowledge to the Salicaceae pathosystem. So the first goal would be to therefore identify the bacterial endophytic communities colonizing the xylem of the genera Salix and Populus through isolation, but also through metagenomic sequencing. Then the idea would be to see the interaction between those isolated bacterial endophytes and the two phytopathogenic bacteria. And to identify those interaction in vitro, and then we want to confirm those interaction in vivo in planta. So let's begin with the culture-dependent method, where xylem sap was extracted with a scolander pressure chamber to collect between 0.5 and 2 milliliters per sample, depending on the size of the stem sample and the time of collection. Colonies were isolated on 4 different solid media, and then 16S rRNA was amplified, sequenced, and then the reads were processed with bioinformatic and statistical tools. That allowed us to see that we had 440 bacterial isolates that were isolated either coming from Salix, here framed in blue, or Populus, here framed in orange. And those bacteria were also isolated on different solid mediums. So you have on BCYE, on KB, on PDA, and on TSA. So if we want to have a better understanding of the core microbiome of Salicaceae that can be isolated from the xylem, we can have a deeper look at this upset plot. So only 5 species are shared by all of the Salicaceae species that we tested. So that are those 5 bacillus. So Bacillus proteolyticus, subtilis, techilensis, toyonensis, and velezensis. Then 9 are common to all Populus species. So to the previous list, you have to add the Bacillus licheniformis and then those 3 Pristia. And 6 are common to all of the Salix species. So again, it's the first list, but you have to add the Bacillus weidmanni. Here you can see the relative abundance of the different bacterial species that were isolated from plant species. So again, here framed in blue, you have the Salix species, and here framed in orange, you have the Populus species. In the Salix species, you can see that we have more the light color, so more Bacillus, and in the Populus, you can see that we have more of the darker purple, meaning that we have more Pseudomonas. And then again, as in the previous slide, you can see in green that we have the 5 that are shared by all of the Salix species. Then in orange and green, the one that are shared by the Populus, and in green and blue, the one that are shared by the Salix. But we wanted to go further than that, and we wanted to be able to confront those isolation results with metagenomic sequencing results. So to that purpose, xylem sap was again extracted by placing the stem in a column and then the column in a collection tube. The DNA was extracted. The library was constructed and sequenced, and the reads were processed. This allowed us to see the relative abundance of each genus that we found in the metagenomic analysis. And by sequencing the V1, V3, and V3, V4 hypervariable region, we were able to gain a complementary picture of the composition of the Salicaceae xylem microbiome. In fact, now we are working to enriching the database on which the sequencing reads are blasted, thanks to the sequence of the isolated bacteria. And while we can see some divergence in the species and genera that we have isolated and the one that we have sequenced, this approach still enables us to better understand the trends and the prevalence of each bacterium that we have isolated. So here, framed in red, you have the genus that were also found in the isolation process. And so now that we have this fuller complementary picture of the xylem microbiome of Salicaceae, we wanted to investigate the interplay between the isolated endophytic bacteria and our two model pathogen infecting the xylem. So to identify the role of these 440 isolated endophytes and their interaction with Xylella fastidiosa and Brenneria salicis, confrontation in vitro were performed, and we selected 40 strains that showed an inhibition halo either for Xylella or for Brenneria. So the drops here are the positive control for Xylella, and in the inhibition halo here, you have an endophyte, and it's the same for Brenneria here. So this one is the positive control, and the halo there is next to the endophyte. To further select among those 40 strains of interest, other in vitro tests were performed. So we did phosphate solubilization, siderophore production, motility assay, indole production, and biofilm formation. And we were able to further select those 40 strains, among those 40 strains, based on the in vitro test score and the frequency of occurrence in isolation. And so we were able to select 15 strains to have their full genome sequenced, to be able to perform some genome mining, and then to transform them to be marked with a fluorescent protein to be able to track them during the inoculation in vivo. So to follow the strain selected in vitro, we wanted to transform them. So here, the Pseudomonas coleopterorum and the Pseudomonas graminis were selected for the electroporation-based transformation with 2 E. coli, one expressing the TN7 Hampshire plasmid and a helper plasmid. And so those selected bacterium competent cell and those plasmid were mixed and electroporated, and then were selected with fluorescence microscopy, and also were checked for insertion with a specific primer in PCR. And then we also wanted to transform the pathogenic Brenneria salicis and the endophytic Erwinia rapontici. And for that, we used a conjugation protocol in which we used the selected bacterium with this time for the endophytic strain, the TN7 Hampshire and the M. neon for Brenneria salicis with a helper plasmid. Again, these strains were mixed and then incubated on selective medium, and the colonies were checked with a PCR primer specific to check for the insertion of the cassette and confocal microscopy. And so the transformation was successful for some of the endophytes. So for the rest of the presentation, Erwinia rapontici will become ER, Pseudomonas coleopterorum will become PC, Pseudomonas graminis PG, Brenneria salicis BS, and we added one already transformed endophyte, which was Paraburkholderia phytophthirmans, which will become PP. And so you can see that the endophyte have the insertion for the Hampshire plasmid, and that the Brenneria salicis has the insertion for the M. neon green plasmid, and it's confirmed with confocal microscopy and on a transilluminator for checking for the fluorescence. Unfortunately, we were unable to transform the bacillus strains that were selected at the beginning of the experiment. But it was time to do some pre-tests to see if those transformed endophytes were able to propagate in tobacco plants and willow plantlet. So we wanted to see which inoculation method would be best. We tested root drenching, leaf drenching, and pinprick inoculation method. And with all of the method and on both tobacco plants and willow plantlets, we were able to find back the endophyte with PCR in the lowest sleeve, in the apex sleeve, and with confocal microscopy. So it was time for the large-scale inoculation in vivo assay. So we performed a co-inoculation with the pinprick inoculation method, and we were going to check for we were going to monitor the symptoms apparition to determine if plants that are co-inoculated with endophyte develop less symptoms than the one inoculated only with the pathogen. So then the propagation and the propagation of both of inoculated bacteria will be monitored, and the plant will be divided in sections to perform the different tests that we want to have. So during the assay, we perform PCR detection on the leaves. And at the end of the assay, we perform a qPCR and visualization with confocal microscopy and re-isolation on selective medium. So here we wanted to investigate the impact of co-inoculation treatment on tobacco plants. So first, we had to establish a visual scale to monitor the symptoms development, ranging from more or less asymptomatic plants to severely wilted and stunned individuals, as showed on those pictures. And so we were able to evaluate symptom severity across all treatment combinations, and they are summarized in this heat map. So on the first box here, you can see the control plants, so the ones that are not inoculated at all, then the ones that are inoculated only with the endophyte. In this second box, you can see the ones that are inoculated with Xylella fastidiosa, first alone and then with the 4 endophyte. And then on the last box, you can see the ones that are inoculated with Brenneria salicis alone, and then Brenneria with the 4 endophyte. We were also able to track the height of the plant at the end of the experiment. And as you can see by the red box, the plant only inoculated with the pathogen were the more severely symptomatic and the smaller one. So it would suggest that there is a possible antagonist interaction, or at least a reduced virulence, when we co-inoculate the pathogen with our chosen endophyte. And we wanted to check that with qPCR. So here, to further understand the observed symptoms patterns, we quantify the pathogenic load in plant tissues. Here, it's still expressed as CQ mean. And so on the left, we have the control group where we, as expected, have no pathogen. Then here in red, we have the Xylella inoculated group, where we observe a high bacterial load in plant inoculated with Xylella alone, so it's that dot. And the co-inoculation with any of the endophytic strains reduce the Xylella fastidiosa population. So with a higher CQ, you have a lower population. So that could suggest a strong suppressive effect on the pathogen. And then here in blue on the right, we see the quantification of Brenneria salicis inoculated group. Again, Brenneria alone reach quite high loads that are sometimes lower with the co-inoculation of an endophyte. So altogether, this result confirmed the antagonistic potential of the selected endophyte against both pathogen in planta. And we wanted to further analyze that with re-isolation and visualization in confocal microscopy. So here, with this table, you can see the number of successful re-isolation from plant tissue. So we observe here that Xylella could be consistently recovered when inoculated. But Brenneria was never recovered by re-isolation. And for the confocal microscopy, we were often able to see Xylella fastidiosa when it was inoculated alone, and less when it was co-inoculated with endophyte, that we were also always able to see. We never found Brenneria salicis in confocal microscopy, whether it was alone or co-inoculated, but we found the endophyte which were co-inoculated with her. And here are some examples of the picture we had in confocal microscopy. So each time in red you can see the endophyte, and in green you can see the pathogenic bacteria. And then as a next step, we want to be able to extend this co-inoculation strategy to a more ecologically relevant model that would be Salix caprea, which is a natural woody host of both pathogen and endophyte. You can see on this symptoms intensity scale that the inoculated plants can show some sign of chlorosis of growth or growth inhibition. But when we look at the heat map, no clear pattern can be found. And also no significant difference was found in the height of the plantlet. So here also, if we look at the isolation, we were able to re-isolate sometimes Xylella when it was inoculated alone, but never when it was co-inoculated with endophyte, that we were able to re-isolate. And for Brenneria salicis, we never re-isolated it when it was inoculated alone, but sometimes when it was co-inoculated with some endophyte. And for the visualization in confocal microscopy, we can see that even if we did not always isolated Xylella, we saw the bacterium in confocal microscopy along with some of the endophyte that were inoculated. We sometimes found Brenneria salicis and the endophyte that were inoculated with her. And again, here are some of the example of the picture that we had in confocal microscopy, with the red one being the endophyte and the green one, the pathogen. So what I would like for you to take home is that we have isolated 440 bacteria from 43 genera and 104 species, that 40 were selected through the in vitro confrontation, and then that 15 were chosen for further testing, for sequencing their genome, for marking them with a fluorescent protein to be able to track them during the in vivo inoculation. That our isolation method was completed with a sequencing of 2 hypervariable regions of the 16 S rRNA to have a fuller picture to what can be found in the xylem microbiome of Salicaceae. And that the co-inoculation of endophyte might play a role in mitigating symptoms development in tobacco plant. And then what is left for me to do is to continue to check for the propagation, multiplication, and localization of inoculated bacteria in the willow plantlet. And we are even doing more assay with endophyte that I did not show you at this time. So yeah, that would be interesting result to complete this assay. And with that, I would like to thank everyone who participated in those projects, those who provided material support or funding, and then thank you for your attention. And if you have any question, do not hesitate.
Thank you very much.
Thanks.
Thank you. And again, apologies for the technical issue, but
no worries, it happens.
great. And going back to, so again, we open now for questions. So anybody has a question, you can raise your hand and speak, or writing in the chat, whatever you prefer, and communicate with Lena.